right, thank you very much for having me. It's a pleasure to be here. I'm very excited to share with you some of the research that's going on. It really is an incredibly exciting time for ALS research. We have successes that we can point to that are here and coming over the summer, and we have a number of programs that follow that to build on those successes. This is a very different landscape than it was just a short 3 to 5 years ago. Not to speak of prior to that. So really an incredibly exciting time. I do have a few disclosures because I'm involved in clinical research. I've worked with a number of companies on advisory boards. I have a fellowship with Voyager Therapeutics developing a gene therapy program, and I've worked as a site investigator on many different studies, including some of the ones that I'll talk about today. This is sort of a mostly thorough, it's hard to be completely thorough and keep up to date, list of drugs that are in the pipeline to be tested and are being tested in ALS. There are many more that are being developed in academic institutions and companies right now that do not make this list. The red ones are the ones that are being tested for efficacy right now. That is, they've made it through some safety testing. We feel like they're safe. We need to know whether they work. And to have that many drugs that are at that point in testing is really remarkable in this field and really incredibly exciting. There are a number of trials that are enrolling, many that have finished the enrollment period and are ongoing, and some that will be enrolling soon. I'm going to talk about a handful of these that I think are informing the field going forward. I'm going to start with a conversation about Edaravone, Radicava. This is the drug that was recently approved by the FDA. There have been a number of really wonderful webinars. ALS Association has put on a few. MT Pharma has put on some. I wanted to actually, many of those have been very practical, sort of about the logistics of the drug and very important, but I wanted to sort of take us through its development in ALS and see how that informs some of the decisions that people are going to be making about this drug. So Edaravone works, as we think, as a free radical scavenger. Now, I would put a caveat on this to say that in the approval documentation from the FDA, they say the mechanism is unknown, because really almost any drug that we work with, including riluzole, we have a proposed mechanism of action, but then it usually takes much more research to know if that's the actual mechanism or whether it's beneficial through some additional mechanism as well. What it means to be a free radical scavenger is that when there's damage to cells or stress on cells, there are these free radicals that are formed. They have an extra electron and they bounce around in a cell and they do damage to proteins and they can cause inflammation. And if we can absorb some of those free radicals, we might be able to reduce the inflammation and reduce the damage to cells. And in fact, Edaravone was approved in Japan in 2001 for care after a stroke, starting within 24 hours of a stroke, given for 2 weeks, to reduce that free radical damage that happens after the stroke has occurred. And then the thinking was that same approach could be used to look at Edaravone in ALS. And so there were some preclinical studies, that is, studies in ALS mice. These are mice that are genetically manipulated to have a human gene that causes ALS inserted, and they develop weakness over time. We've been using these mice to inform our research about this disease for 25 years, and we do a lot of studies with potential drugs in them. And what the researchers found was that if they treated these mice with Edaravone, they could preserve motor function. So you'll see in this graph, the red line is a high dose of Edaravone treated mice, and they have a slower decline in their function over time. This next graph, these 3 bars here in the middle, show that it reduces markers of oxidative stress. Now, these markers of oxidative stress could indicate that the drug is actually working through its proposed mechanism. And that was the importance of looking at those. This is, this middle graph shows some of that. It's not statistically significant, but I'm going to come back to this point in humans in just a moment. And the graph all the way on the right-hand side shows that there is loss of motor neurons in the transgenic mice, in these ALS mice, and you can rescue that, at least to some degree, with the drug. Now, there was a study of Edaravone looking at whether it can prolong the survival of these mice. The drug was given at the time that the mice developed symptoms, and I'll come back to why that's important in just a minute. It did not show that Edaravone prolonged survival in the mice. The reason it's important to say that they were treated at the time of symptom onset is that is an incredibly high bar. These mice become sick, they develop weakness, and they have a very, very aggressive course that mimics ALS in some ways, but we have to acknowledge that this is just a model of the disease. It is almost impossible to find a drug that reaches this very high bar of treating disease after it started in mice. So I think we have to take that piece of information with a grain of salt. But mostly we have really encouraging data from the preclinical models. And so the investigators then went on to develop this drug in humans. And they did a typical drug development, moving through the phases of development. The phases are phase 1, 2, and 3. Now for Edaravone, because it was already approved for another indication, phase 1 wasn't required. Phase 1 is really the first time we give a drug to humans. We sort of give it to a small group, we give it in a single time point, and we see whether there's any catastrophic problem with the drug. That had already been done, and we didn't need to redo that. So the investigators jumped in at phase 2, which is to say, what's the safety over a longer period of time? What kind of dosing should we look at? And can we set the stage for our effectiveness trials, which are phase 3 trials? And there were 2 phase 3 trials done. So I'll walk you through some of that evidence. Now, in all of the trials, the treatment period was 6 months long, and the dosing regimen was the same. The drug was given for 14 days daily through an IV, and then 14 days off. And then the subsequent cycles, we'll call them, were 10 days out of 14 on, and then 14 days off. And that's the same way that it's been approved, I should jump ahead and say. The primary endpoint in these trials was change in the ALS-FRS-R over those 6 months that they were treated. And to remind everybody, the ALS-FRS-R is the ALS Functional Rating Scale revised. It's a 12-question questionnaire. It goes from 0 to 48 because each question has a 0 to 4 scale. So this phase 2 trial that was done was actually an open label trial. It's the only trial that I'm going to talk about that was an open label trial. That means everybody received treatment. There was a period of time that people were observed before they were treated, and then they were observed after they were on the treatment. There were very few side effects, a little GI upset in one patient, maybe some mild lab abnormalities. It wasn't clear they were related to the drug. Very safe, which is the experience of this drug in stroke treatment in Japan as well. There was a hint that there might be efficacy here, even in this open label trial, in that there was a decline of the ALS-FRS-R of 4.7 points, 4.5 points over the run-in period, and only 2.3 points in the subsequent period when they were treated. Now, we are always cautious about interpreting this kind of result in an open label trial, but it is very encouraging to see a result like this and led the investigators to move on into a larger trial. So they moved on to a placebo-controlled trial, a phase 3 trial design, again, similar endpoint, 206 patients in this trial. The criteria were fairly tight, and this sort of becomes important for the conversation of sort of how we generalize the trial, and I'll get to that in a moment. But the reason the criteria were fairly tight in this is they were aiming to study people who were early in the disease where they thought they could have a discernible impact in the trial. It was about trial design, not necessarily about biological premise. And so that's an important point. People had to have had symptoms of ALS for less than 3 years. Their vital capacity needed to be over 70%. None of the questions on the ALS-FRS could be lower than 2 points. And they needed to work and live independently. There's a Japan severity scale that we don't tend to use here, but essentially it meant that people had to be able to live and work independently. And they could either be on or off riluzole, but had to be on a stable dose if they were on it. Fairly typical criteria, actually, for many of our trials. The trial did not meet its overall endpoint. There wasn't a statistically significant difference between the treated and the untreated patients, the placebo and the treated patients in the trial. Having said that, the investigators looked at a few subgroups, and they divided people into what we call our Ellis-Scorial criteria. And those Ellis-Scorial criteria have a somewhat confusing name of possible, probable, and definite ALS. This is not about how likely the diagnosis is. This is about how many regions are affected by the disease. And when they began to divide individuals in this way, they found that actually some of the groups may have responded somewhat more robustly. But there was a trend in all of the groups, so that was in the right way. That was sort of a trend towards slowing. What I mean by that is it could be just due to chance. You can't statistically prove that this is absolutely could only be due to the drug. But the data shows, that data point shows that in this case, it's possible that there was a beneficial effect of the drug. So that was encouraging in some respects, but not enough to sort of be the thing that would lead to approval alone. In the meantime, there were trials looking at biomarkers. And biomarkers are important in this case because they give us a biological premise. They say, is the drug hitting its target? Is it doing what we think it should be doing? Is there a reason to think that there is an effect in the body that could be leading to the effect we think we might be seeing in the clinical trial? And this colorful graph shows uric acid. Uric acid is, in fact, an endogenous sort of inside of us antioxidant. And it looked like, it's a little hard to see from this graph, but it looked like almost all the people that were treated in this separate trial just to look at the biomarker effect, looked like almost all of them had an increase in uric acid. So that would be an increase in this endogenous antioxidant, and that could be taken as a really beneficial sign. There was also a look again at this measure of long-term oxidative damage called 3-nitrotyrosine. It showed up in the animal studies. It wasn't super robust there. I said I would come back to it in the human trials. Here we see a graph showing a substantial decline in 3-nitrotyrosine in those who were treated with Edaravone, giving us the biological premise to say this seems to be working the way that we think that it does. The evidence from the phase 3 trial and the biomarker studies encouraged the investigators to go on and design another trial. This was a smaller trial than the previous phase 3 trial. There were 137 participants. They were randomized 1-to-1, so half the people got placebo, half the people got study drug. That, from a statistical perspective, gives us the most power to see change. It's the design that requires the fewest participants in a trial. The dosing regimen was the same, and the inclusion criteria were similar, but even more strict. So people had to be within 2 years of symptom onset. They had to have a vital capacity over 80%, and they also had to be taking Riluzole. And these were all things that were done to create a group of people enrolling in this trial that were as similar as could be to one another. The reason for that is it cuts down on the noise in the data. Any heterogeneity, any differences, variability in the people that are enrolling in a study make it harder to see the signal from the drug. So this was purely a statistical choice at this point to try to mimic and build on the successes that were seen in the previous trial. The results of that trial were positive. That is to say, the people who received placebo had a drop of 7.5 points in their ALS-FRSR over 6 months. The people who received Edaravone had a drop of 5 points. So a difference of 2.5 points over 6 months, a 33% slowing of disease. That's what that trial showed. That is what led to the approval in Japan, as well as in the US. I think in addition to that, few side effects were seen. Many of the side effects that we see listed in the FDA prescription information probably are symptoms of ALS, to be honest. But they listed everything that showed up, even slightly more commonly in the treatment group than in the placebo group. So we see things that come up like respiratory failure or disorder, and that probably is not the drug. That would have shown up as an important signal. Contusion, gait disturbance, headache. I think, some bruising at the site of the IV was seen, headache. So potentially side effects. The other thing is they did see a few allergic reactions, more common in people who have a sulfite allergy. Important to say that sulfite, not sulfa drug. Sulfite is in red wine. Many people know they have this because they have an allergic reaction to red wine. So the FDA actually went to MT Pharma and asked them to last summer to put in a new drug application based on the evidence in Japan. Usually the FDA requires 2 positive phase 3 studies in order to approve a drug. Usually one of them has to be done at least in part in the US. And so this approval really was different than the standard pathway. And I think that really sort of speaks to the organized efforts of people with ALS, caregivers, much of it driven by the ALS Association, to say to the FDA, to lawmakers, to researchers, listen, what we want is if something looks safe and looks like it's doing what we think it should be doing, don't hesitate. And so I think it's just important to sort of note that it appears that those voices are heard, and that's really encouraging. The major question that I think is in front of us now and is generating a substantial amount of debate amongst researchers and people with ALS and caregivers and the public and anybody who cares to think about this is, can we generalize the results from the trials that were done in people with early ALS and rapidly progressive ALS to everybody with ALS? And I think there are a few ways to think about this. There are kind of two major things at play. One is to say, possibility one is, maybe Edaravone has some biological reason that it would only work in people early in the disease or would only work in people with rapidly progressive disease. And in fact, we might say, well, maybe there's more free radical formation, more neuroinflammation in people with rapidly progressive disease, maybe more early in the disease. So we could figure out a biological principle. And that's a possibility. We don't have any proof of this. The fact is that free radical formation and oxidative damage and the inflammation that comes with that appears to be present in people, from everything we can see, appears to be present in people with ALS across the time that they have the disease, throughout different subtypes of the disease. So from a biological perspective, it seems reasonable to think that this drug has a target in most people, if not everybody, with ALS. The second possibility is that these restrictions that were put on the trial were really put there for very trial design specific reasons. They were done to create this homogeneous group of people going into the trial and to boost our signal to noise ratio. And they did that, but the results then are generalizable to the population of people with ALS. And this is where the FDA came down, because this drug is approved for all people with ALS. Now, there's no proof right now that it works late in the disease or for people who have more slowly progressive forms of the disease or wouldn't have fit the criteria for the trial in another way. But I think in the absence of evidence, the FDA took the broad approach that it's approved for all people with ALS. I think ultimately, whether this is the right drug for somebody to try is going to be a decision between a physician and a patient, but hopefully looking at some of the evidence and thinking through some of these questions now may inform those conversations. And I'm happy to answer questions about it later. I think the other thing to know is that for people who are in trials, most trials are going to allow people to be on Edaravone and be in the trial, either for trials that are going on now or trials that are starting, but it will be trial by trial specific. And so it's important to ask and find out from your trial team whether there's any conflict. Sometimes if there's another drug that's in the same pathway, working on neuroinflammation or working on antioxidants, there could be a potential safety conflict, and that's something that would have to be managed for that trial independently. So there's been, as I said, I'm not going to spend a lot of time on this because I think there's been a lot of really informative conversations about the implementation of this, and you can look for webinars, I think, on the ALS Association site, the Northeast ALS Consortium website, and MT Pharma itself. But we're sort of, I think at every center, we're thinking about how do we reach out to all of our patients, not just the ones who are knocking down our doors to get the drug, but also people who may not be informed about it, may not be as connected to these information sources. So we're making a big effort to sort of get the word out to our individual patients. We're going to have to wrestle with insurance prior authorizations, and we don't know how insurance is going to handle this. There's a lot of work going on to try to make sure that goes well and that insurance does cover it. And then, I think we're working out these infusion schedules. This is going to be a big sort of change for how we do this, but home infusion is certainly a possibility. Infusion centers have a lot of capacity as well. And the last thing that we're working through is IV access. So we can do this through a peripheral IV. We can do it through potentially a port. There may be upsides and downsides to that. That's a sort of a drum that goes under the skin. Or we could do it through what's called a central line, which has a sort of a plastic piece that comes out from the skin and can be connected to. And those are all conversations that we're going to have and make and sort of find the best way going forward. So very exciting to have a second drug to treat this disease. And I would also just pause to say, if we look at a disease like multiple sclerosis that has a number of quite effective treatments, still a lot of research going on to improve those, if we look back to their first treatment, it was onerous to give, it had a lot of side effects, not well tolerated, it was an injection medication, and not so effective. Some effect, but not so effective. And then there were more. They were easier to give. The schedule was more tolerable. They had less side effects. They were more effective. And then fast forward another decade, there were oral medications. They were more effective, fewer side effects still. And so I think there is a paradigm that says we start where we are, and maybe the infusion schedule is not exactly where we want to end up, but we can build on what we have. We can build on our successes. So incredibly important, I think, to have that as an option. I'm going to talk now about Masitinib, which is another phase 3 trial that recently completed, and the results have been presented at least, but not published. This is a trial that has been led out of France, and the trial was done in Europe. Masitinib is what's called a tyrosine kinase inhibitor, and that's a receptor on the surface of inflammatory cells. So initially it was noticed that Masitinib inhibits degranulation of mast cells. So mast cells are these cells that circulate in our body, and they contain histamines, and they release those histamines. That's called degranulation. Histamines are these things that make us sniffle and sneeze and give us skin rashes, and it's why we take antihistamines to try to get rid of some of those symptoms. These are really important for asthma attacks and allergic attacks, and so it's being studied in those conditions as well. But it was also discovered that these are really important receptors on monocytes and microglia. Microglia are the immune cells of the brain. We know they're activated in ALS, and so it's sort of a potentially on-target therapy for one of the problems that we know is going on. So the trial in Europe included around 400 people. There were 3 arms, a high dose, a low dose, and placebo. They were randomized 1 to 1 to 1. The outcome was looking at the ALS-FRSR change from baseline to week 48. So it was a year-long trial. And there were secondary outcomes looking at change in vital capacity, looking at something called the CAF score, which is a statistical approach to analyzing data. There was an interim analysis that was released in a press release about a year ago. It essentially said that the interim analysis was positive. It didn't give a lot more information than that. There is something that we talk about called a p-value. That p-value is sort of how we think about what we call statistical significance. So what are the chances this could just be due to random variation? And just to give you the number, we look for a p-value of less than 0.05. And so this met that measure, but we didn't really have much information about what measure they were talking about. The data was presented recently at a European network of clinical ALS researchers meeting. And again, this is the data as it was, a summary of the data as it was presented, so they reiterated the design. They talked about the primary analysis. So the primary analysis is when we design a trial, we have to say, here's how we're going to analyze the data. This is how we're going to decide whether the drug worked or didn't work. And what they presented was that their primary analysis was the absolute change of the ALS-FRSR at 48 weeks, only comparing those who got high dose to placebo, and only comparing those who had a less than 1.1 per month point change on the ALS-FRS prior to treatment. So a subgroup of the people who were in the trial, and that's somewhat unusual, we don't have any more information than that because, as I said, it hasn't been published, but that is an unusual thing to write in as a primary endpoint, to enroll 400 people and analyze only a subset of those people for your primary endpoint. So I think it's something we have to learn more about. The endpoint comparison showed a slowing of the ALS-FRS of about 25%. So over a year, there was a drop of 12.6 points in the placebo group and only 9.2 in the treated group. This is the high dose and only people with a certain change in their ALS-FRSR prior to treatment. There were other supportive analyses that had to do with quality of life scales, vital capacity scales, and the safety was reported as acceptable. So encouraging results. There are some things I think we need to understand better. I think we're sort of waiting to see a publication that gives us all of the data, and we're waiting to hear what the next steps might be for this. The next trial I want to talk about is really an innovative trial, and I think one that is the canary in the coal mine for a whole new area of research, not only in ALS, but in many, many diseases. And this is, the drug is called BIIB067. There'll be a quiz at the end. It's what we call an intrathecally delivered antisense oligonucleotide. So let me walk you through that. Intrathecal just means we put it into the spinal fluid. Antisense oligonucleotide refers to the kind of drug that works, and we call these ASOs, by the way. ASO is less of a mouthful, so I prefer it. Antisense oligonucleotides, or ASOs, are man-made pieces of DNA. And in this case, it is complementary to something called SOD1. So the way that our cells work is we have DNA, and the DNA is sort of the blueprint for everything that goes on in the cell. The DNA makes RNA, okay? RNA makes its way out of the nucleus into the machinery of the cell. Okay? RNA is translated into proteins, and proteins are the building blocks of the cell. In a certain form of ALS, known as SOD1 ALS, there's a mutation in the SOD1 gene. So the DNA is wrong, miscoded, there's a problem. This is passed down in families. It's a dominant trait, so half of the people in a family are at risk for developing this because half of the people would inherit this gene. It's dominant, so you only need one gene. And it's about 2% of ALS. If there's a mutation in the DNA, then the RNA that's made is wrong, and that means the protein that's made is wrong. And this protein folds incorrectly, and it gets it accumulates in the cell and it gets caught up. And that causes the disease, we think. This little piece of DNA that we make, synthetic DNA, antisense oligonucleotide, we deliver to the cell. It binds to the bad RNA, to the wrong RNA, that mutated RNA. And the cell then recognizes it through endogenous mechanisms. The cell knows how to do this. It recognizes this mismatch of DNA and RNA, and it destroys it. So by destroying the bad RNA, it means there's less of the bad protein. That means there's less of that protein to accumulate, and that means we might ameliorate the disease. And in fact, there's evidence of this. So preclinical data in animals, and I'll show you some of this, show that we can knock down the protein, the bad SOD1 protein, and suggest that this is probably safe and that this is potentially effective in treating the disease. Not only that, it says that we might be able to measure SOD1 protein in spinal fluid as a biomarker of what's going on in the brain and the spinal cord. So we know we're hitting our target, we know we're doing what we think we're doing, and gives us biological premise to believe our clinical results. We have good natural history data about SOD1 ALS. This is a rare form of an uncommon disease, and therefore the more we can do to understand the course of the disease and have what we call historical controls to compare our data to, the easier it will be to show efficacy. Not only that, but there actually has already been one trial of an antisense oligonucleotide in people with ALS, and I'll just talk about that briefly in a moment. Just to go back to the delivery, there are two major ways to deliver things intrathecally into the spinal fluid. One is into the brain, because we have sort of cavities in the middle of our brain that contain spinal fluid. And the second way is in the lower back, where we do things like spinal taps, but here we could inject drug. And this is being injected through the second route into the lower spine. And it seems to go throughout the central nervous system quite well. So in ALS rats now, not ALS mice, but ALS rats, this ASO has been used, and we can demonstrate that it reduces SOD1 protein in the brain and spinal cord. And that's important because that's where we want it to act. But importantly as well, we can see that change in the spinal fluid. So the change that's going on in the brain and spinal cord is reflected in the spinal fluid. And that means we have a biomarker. That means we can tell if our drug is getting to its target, which is the first step, during a trial. And that means we won't spend time developing a drug that's not getting where it should go or not doing what it should do, because if we don't see the biomarker change, we'll know that we have a problem in the people. It guides our development and is incredibly important. We also know that a previous trial that was done, small number of centers, different antisense oligonucleotide, was safe, was well tolerated, and really set the stage for doing this. In fact, it was the same company then called ISIS Pharmaceuticals, now called Ionis Pharmaceuticals, for fairly obvious reasons, and now in partnership with Biogen. It was actually right after a Saturday Night Live skit that they changed their name. That's true. That's a true story. The current trial that's going on is similar to that phase 1 trial that was done, but it's actually a new antisense oligonucleotide. There was a fairly long pause between that phase 1 trial and the trial that's going on now. And the reason for that pause is that the chemistry for building these molecules improved dramatically over the course of doing that trial. And so we knew we could get better, the broader community, the company, knew that we could get better knockdown of SOD1 with a new chemistry. And I think took the time to really get that chemistry right and go back into trial. Again, it's being given by intrathecal bolus. The dosing regimen slightly different than I think they had planned for previously because of the new efficacy of this, expected efficacy of this antisense and how long it can stay around. So it's only dosed about once a month. The plan for developing this is kind of, you have to, anytime you change the chemistry of a drug, you have to go back to the drawing board. So it's really a phase 1 and then phase 2 trial kind of mashed into one. And so it's given first as a single dose and then into multiple doses. And there are things like travel programs and more sites to try to make this, and sites in Canada and Europe, to try to make this more accessible to more people with SOD1 ALS. The primary objective here is really to look at safety. And also something that we call pharmacokinetics, which is just where do we find the drug in the blood? Do we find it in the spinal fluid? How long does it stick around? And also to look at the effect of the level on SOD1 protein in the spinal fluid. That's our biomarker that we talked about. It's called a pharmacodynamic biomarker or a target engagement biomarker, specific kind of biomarker, but really important to understanding this. And then the exploratory outcomes are really on the clinical things, the things that we ultimately want to know. Does it slow the disease? Can we see an effect in the muscle, in the strength, in the breathing, and on the ALSFRS-R? This is a diagram of the trial. These first cohorts, cohort 1, 2, 3, and 4, are what are called single ascending dose, SAD. Maybe not the best name. Trumped only by the next phase, which is called multiple ascending dose, MAD. So we have a SAD study and a MAD study. And these are sometimes separate studies, but in this case, they overlap. And what we do in the single ascending dose is we start at a low dose, we give a single dose. If it looks safe, we move in a new group of people to a higher dose. If that looks safe, we move to a higher dose. And they're coming to the end of this SAD study now. They're in the last cohort. The MAD study, the multiple doses, has now also begun. And they also have an expanded access phase to this so that people who have been in the study can continue to receive open label medication, which is really a wonderful thing for the people who have participated. So I think I would say, just in closing about that trial, this is, I think, the first of a number of approaches to affect gene products. There are gene therapy programs that are in preclinical phases to use a virus to deliver a similar kind of a message to the cell, but one that's given once and then expressed throughout life. This is work that's going on at Voyager Therapeutics. So you treat people once and you can affect their sort of genetics for the rest of their life, potentially, or at least what's expressed. There are other antisense oligonucleotide programs that are in the works at Biogen, so for C9 ALS, another form of ALS. And we now know increasingly that there are gene modifiers for ALS. And so this may be something that comes out of kind of genetic forms of the disease into sporadic forms of the disease, and we may be able to put in a genetic modifier to slow the disease or knock down a genetic modifier that's hastening the disease. So I think this is a technology that is a harbinger for a huge paradigm shift in medicine, but ALS is really at the cutting edge of it. Tirasemtiv is another drug that's being studied now. It's in a phase 3 trial now. These are a summary of results provided by Jeremy Scheffner who was here for a long time, of a prior trial. And I'm going to just sort of walk you through what we know about tirasemtiv so far, and then tell you a little bit about the design of the trial that's going on now. So hopefully people can see the cursor. This line in the middle, that vertical line, that line is sort of the line of no difference, okay? Any point that's over here to the right of that line favors tirasemtiv. Anything that's to the left of the line favors placebo. And we have a number of outcome measures listed here. And then we've got green dots and red bars. So the ALS-FRS-R is at the top. There's a green dot. It's just maybe slightly to the left of that line, but largely on the line. And those red bars cross the line. What that means is there really isn't a statistical difference in the results of the ALS-FRS-R for people in this previous trial. We didn't see an effect there. If we come down to the predicted SVC, we see that green dot lies to the right side. That favors tirasemtiv. People who received tirasemtiv had a slower decline in their vital capacity. The red bar, those red lines attached to that green dot are the error bars. They don't cross the midline. There's really very little chance that dot is in the wrong place. If you come to something like MVV, which is minute ventilation, there was a lot more variability there. SNP is a sort of a test of how people can breathe in through their nostrils. Grip fatigue. So this favored tirasemtiv, but look how big those red bars are. Really big red bars. That means there's a lot of variability in the outcome measure. This speaks to why we're constantly trying to reduce variability in our trials, because even though that green dot is on the correct side, we can't tell for sure where it should lie. Could actually be anywhere in those red lines. And the less variability there is, the more we can sort of say for sure that we've had an effect. The last point here, which also has variability but lies entirely on the favors tirasemtiv side, is quantitative strength testing, something called handheld dynamometry. And so not every measure showed a benefit, but there were at least two important measures, breathing and strength, that did appear to show a benefit. And that led to a second phase 3 trial, which is going on now. Without spending too much time on this, there was an open label phase where people received the drug. And there are some people who have side effects with this drug, nausea or dizziness. And if they had those side effects, they then weren't put into the trial. There are 3 different doses that are being tested in the trial against placebo. People are treated for 24 weeks and then the primary outcome, but they're actually treated for 24 more weeks, so it's a year-long trial. And at the end, people are randomized to either continue drug for the last month or to come off of drug for the last month. And that's to see if there's a prolonged benefit of the drug even after people stop it. And that comes out of data from the previous trial as well. This is a large trial. It's a trial that's meant to understand for certain whether this drug works, and we're looking forward to the results. The people should be sort of ending this trial probably in the fall, and we'd look forward to results, hopefully next winter. But there is a long period of time where we have to check every data point, lock the data set, and do some analysis before those results are available. The next trial I'm going to talk about is a trial of a technology called NurOwn, which is mesenchymal stromal cells secreting neurotrophic factors. It's a kind of stem cells, and these are autologous stem cells. These are stem cells that come from the person that we're treating, from their own bone marrow. They're taken from the bone marrow, they're grown in culture, that means in a petri dish, and then given back to the patient in the muscle and into the CSF. And this trial, sponsored by Brainstorm, was done in 48 patients at 3 different centers. Ours was one of the centers. Each center treated 16 people. Here, the randomization was different. It was 3-to-1. So for every 3 people that received the cells, there was 1 that received placebo. And the reason for that is that the primary endpoint here was really safety, and the more people we have exposed to the actual study treatment, the more we can learn about safety. But having the placebo there allowed us to have more certainty about those safety signals and also explore efficacy signals. The cells were given in 24 injections intramuscularly, 12 into the right biceps, 12 into the right triceps, and then also intrathecally, as I explained before, into the cerebrospinal fluid in the low spine. People were enrolled in the study. We monitored them for a few months prior to treating them with the transplant. Partway through that monitoring period, we did a bone marrow biopsy, took bone marrow from the hip. Those cells were then, as I said, grown in a petri dish. They're grown and differentiated, and what that means is they're expanded, so we have more of the cells, and they're also induced to secrete neurotrophic factors. Now, neurotrophic factors are proteins that are given off by cells that are like fertilizer for motor neurons. So that when we put these cells back into the body, they are secreting more neurotrophic factors than they were before. And the idea behind this therapy is that these neurotrophic factors and the cells themselves support the motor neurons, create a better environment for the motor neurons, and may even reduce inflammation. It is not that they replace motor neurons or become motor neurons themselves. That's an important thing to understand, I think. And that's true of all the stem cell trials that are going on now. So the trial showed no deaths. There were no treatment-related SAEs. This means serious adverse events, something that would put someone in the hospital. We didn't see any adverse events that led people to drop out of the study. And the common adverse events were mostly mild and moderate. Many of them happened at approximately around the time of the transplant. Things like back pain, headache, that were probably related to the procedure. Somewhat more common in the treatment group, but also occurred in the placebo group. Now, I'm going to walk you through this slide because it's a little complex, but this is some of the efficacy data, some of the data about effect. So all the way on the left-hand side of this graph, you can see two bars of blue and red that are sort of light, faded blue and red. Okay? In the pre-treatment period, this was the slope of the ALS-FRSR, and the blue is those who are fated to have the treatment with the cells. The red is those who are fated to have treatment with the placebo. As you can see, the rate of decline was about the same. It was around 0.7, and it was about the same in both groups, which we would expect because they were randomized. In the period after the treatment, those who received treatment with the stem cells, represented in blue here, had a slowing of the slope of decline. We did not see that in the placebo group. We see that slowing at 2 weeks, at 4 weeks. By 8 weeks, it really is we don't see it anymore. So it appears to be a transient effect. Not surprising for this technology. In fact, the truth is, going into this study, we did a single dose study, just as I talked about with a SAD study, a single ascending dose study. We did a single dose study because we didn't know how long an effect might last. But everything that's been done with mesenchymal stromal cells has said, this may be a transient effect. You might need multiple treatments. And that was really the plan sort of all along. So not surprising to see that. We also plan to look at a group of more rapid progressors on the principle that we may see a more robust signal in this group. And in fact, what we've done is take people who have a progression that's at the sort of midpoint or faster, and that's the group that's represented here. And if we look at that same faded blue and red bar all the way on the left-hand side of the slide, we see that the progression prior to treatment was faster. That makes sense because we've taken out all the slow progressors. And here it was a little more than 1 point per month, actually a little faster in those who were ultimately received treatment with the stem cells than in placebo. And in this group, we saw a more durable, more robust slowing of the slope of ALS-FRSR after the treatment period, only in those who received treatment. Very encouraging. Again, this is a smaller group of people than the overall group, so it needs to be confirmed, but promising. What I haven't shown you is some data from CSF showing that neurotrophic factors increased and that some markers of neuroinflammation decreased in those who were treated with the stem cells, but not in placebo. So that gives us a biological premise as well. We assess the safety and tolerability. We assess this biomarker. We have a hint of efficacy. It looks like that efficacy will require multiple treatments, which has been the plan. And this sets the stage to move on to a larger trial with multiple treatments, probably between 8 and 12 weeks, probably focusing on those who had the most benefit, which is those with the more rapid progression. And the last one I'm going to talk about today is NP001, which is a phase 3 study that's going on now, building on a phase 2 study that was done previously. So NP001, produced by Neuraltis Pharmaceuticals, sponsored by Neuraltis Pharmaceuticals, is sodium chloride, not sodium hypochlorite, which is bleach. Different effect. We shouldn't try this at home. There actually was a previous iteration of this drug that was sold overseas, never approved in the US, that wasn't pH balanced and caused a lot of sclerosis to veins, and this is a pH balanced form of it. Within monocytes, macrophages, and microglia probably, this chloride is converted to something called taurine chloramine. And this is sort of a regulator of an inflammatory pathway in cells. And by regulating that pathway, we can move these cells more toward a homeostatic mechanism. So again, it's a very specific anti-immune or anti-inflammatory treatment. Really, we think targeting a very specific pathway that we think is relevant in this disease. The previous study was a large phase 2 trial, 136 patients, so similar to the size that Edaravone was, pretty large. And the dosing mimics something called IVIG, which occasionally people in the diagnostic phases of ALS receive because it's relevant for some of the disease mimics that we think about with ALS. It's given over 5 days in the first month, and then over 3 days in subsequent months. And in between those infusion cycles, there's really nothing that people have to do. So it's a lot like Edaravone in that way as well, except the cycles are shorter, the on cycles are shorter. The previous study had 3 arms, a high dose, a low dose, and placebo, randomized 1:1:1. Unfortunately, it did not meet its primary endpoint of showing slowing of the ALS functional rating scale, which is what we had hoped for. But there was what we call a post hoc analysis. This means after the trial's done, we look back at the data and we think, sort of scratch our heads and say, is there some signal in here that we would see if we analyze the data slightly differently? And what they did was to look at how many people had no change in their ALS functional rating scale over the entire 6 months. And there was a higher percentage of people in the placebo arm than in the low-dose arm, and a higher percentage in the low-dose arm than in the high-dose arm. So there was it looked like those who received treatment were more likely to have no change in their ALS-FRSR, and it looked like there was a dose-response curve that maybe you could draw. We have to be careful with post-hoc analyses because they're statistically fraught with some potential for bias, but still, I think it's informative, as we've learned from Edaravone, to look at these analyses. They did a second subgroup analysis, and this was to try to identify people that had higher inflammatory components, because this is not one monolithic biological thing going on that causes ALS, but probably multiple different things that are going on, and maybe in different ratios in different people, maybe completely different in different people with this disease. So there's a molecule called CRP, or C-reactive protein, that is a measure of inflammation in the body. And what the investigators did was to go back and look at those participants who had high CRP. And if you look at those who had a high CRP, defined as above the average CRP for that study, if you look at that group, then you see this dose-response curve where the people who received placebo had the fastest progression, the low dose had the second fastest progression, and the high dose had the slowest progression over the course of the 6 months. So this was a second piece of evidence that maybe there is something biological going on with this trial, and maybe it does deserve yet another look. So the current trial that's going on, which is still enrolling but coming to the end of the enrollment period, is pre-selecting participants with an elevated CRP. Has to be above the midline of the previous trial, because that's where the effect was seen most robustly in the prior trial. The idea is to enroll 120 participants, potentially a little bit more than that, actually, to increase the chances of seeing it. It's a 1-to-1 randomization this time. That gives the best statistical power. Only the high dose versus placebo, 6 months of treatment, and the primary endpoint is the change in ALS functional rating scale. And I'll tell you, having enrolled for this trial, it's been very challenging. Half the people who screened for this trial have not qualified, simply because of their CRP level. And that's a difficult thing to do, I think, as a researcher, and a difficult thing to go through as a potential participant in a trial. I think the thing that made it easier, at least for our group, is that based on the data that we saw from the prior study, this is the group that's most likely to respond. And we don't want to put people into a trial if they're unlikely to respond. Better to know that and then move on to other potential opportunities. So again, we'll anxiously await these results, probably winter, spring, once we get all the people through the trial and data cleaned. There are the sort of work that went into the studies that I've presented here is far too grand to put onto one slide. But I want to say that many of these trials were run through the NEALS network and took many investigators, many participants at all the different sites. Terry Hyman Patterson, Jonathan Glass are the current co-chairs of Northeast ALS Consortium. Merit Sikovich and Jeremy Scheffner were co-chairs for many years prior to them and have been important. I work at the MGH Neurological Clinical Research Institute and at the clinic, and our team there is only partially represented here. It's an incredibly dedicated group of people, and we couldn't do any of this work without a huge collaboration, both within our center and with other centers. And the last thing I would say is that the ALS Association has made much of the work here, much of the work I haven't taken time to talk about because I can only fit so much into one presentation possible. Though the whole presentation has been about research, I also just want to take a moment and say that what Barb talked about earlier about a focus on advocacy and on care is also very dear to our hearts. And ALS Association has supported us in many novel care approaches, telemedicine, partnering with home care organizations, trying to understand the multidisciplinary clinic and how to optimize that. And so that's hugely important, I think, in improving our approaches to ALS care as well.
Jim, can I ask you to address one thing that is coming up more frequently? As drugs come through the system and are approved by FDA, and we might have another drug that gets approved that's based on clinical trials in other countries. And let me just footnote that by saying that one of the things that FDA has told us is that the work that Jim and others did on an FDA guidance document to help the FDA has made a big difference in them supporting some of the work, for example, like Radicava. That made a huge difference in their approach to supporting that. But we are getting a lot of folks living with ALS and their families who are saying, I can, through another source, I can get Radicava right now sent to me from overseas. Why is that not a good idea to do?
Yeah, so first of all, I think my major point here is going to be anything like this you want to obviously discuss with your treating physician. And I'll talk about, I have to say, many of us are still catching up to these kinds of things. We've talked with the company about this because the company says we don't think this is a good idea. They make the drug, Radicava. It's got a different name in Japan, Radicava, and they make the drug that's coming to the US, Radicava. What the company has said is that that in order to ensure that a person is getting a drug that's approved, that has been quality tested, that we know about, there needs to be a sort of queryable chain of command, or chain of custody, I should say, not chain of command. But that chain of custody is really important because it's what guarantees that the drug that leaves the factory is the drug that goes into the patient, the person. And that's where it gets, that sort of chain of custody is where it gets tricky. So there are some organizations that are finding ways to move drug around the world, but it's, at least for me, it has been not possible to kind of verify what's happening to that drug in the meantime, which raises some red flags. Yeah, it's a good question. So yeah, the legality of it is, I wasn't going to go into this. The other part of this is that there are sort of complex sort of laws about this, both for physicians and nurses, and then also kind of on higher regulatory rules. And we're at risk of sort of practicing outside the laws of medicine if we are directly involved in any of this. And so it's a challenging situation.
Which leads to another question that we are getting a lot, which is around what's happening on the cannabis front.
Wow, it's like running right into the fire with you. Yeah. Again, this is not the sort of legalities around this are not my specialty. But
right, I'm actually talking about you as a physician and what you're seeing.
Yeah. So this is we could have at least an hour-long symposium, but we should have somebody with more expertise up here to talk about cannabis. So state by state, things vary. So Massachusetts, there is medical marijuana, and actually recreational marijuana has been approved, I think, and yet not really rolled out. But that's at odds a bit with the federal rules, and I think that is sort of being worked out. There are state by state various ways that physicians interact with medical marijuana. And different ways that patients have to interact with medical marijuana as well. So I think it's just this one you have to kind of go to your treating physicians and see. The second question is, what data do we have about the effect of medical marijuana? And we don't have a lot of scientific data on that. There are anecdotes, and it's difficult from my perspective to kind of get to the bottom of this. I would actually love to hear other people's. It's difficult without data to really know what to say about this. There's talk about CBD and THC and the components of these things, and in what appears to be a largely unregulated industry, it's just very difficult to kind of give strong advice, especially to a group like this. But even one-on-one, it's difficult. But thank you for bringing up the very thorny topic. That was great.
Well, I have, and I'll just say this, I have great trust and faith in Dr. Berry and his candor. And so if we're going to have a thorny topic, I'd rather talk to someone that I have confidence in terms of how they're going to respond to it, to give us accurate, truthful information.
Thank you.
There are no alternative facts here.
Is the Edaravone the same thing as Radicava? Is that a different drug or?
Thank you for asking this. I'm sorry that I meant to clarify this early on. Every drug has at least two names. It has a generic name, and then it has a trade name. And it may have more than one trade name. And that's the situation we're in here. Edaravone is the generic name. Radicava is the trade name in the US. So the companies usually give you sort of a shorter, snappier name when they make the trade name so that people can remember it. So Edaravone and Radicava are the same thing. In Japan, the drug is called Radicut. That's also the same drug, different trade name. And so companies do this sometimes. They name a drug a different thing in different countries, depending on what plays better.
Oh, I didn't know we were looking. James,
Yeah.
I missed it. The current SOD1 ASO trial, is that being done by the same group?
So the question is the current antisense oligonucleotide trial for SOD1 being done by the same group that did the previous phase 1? And the answer is essentially yes. So ISIS Pharmaceuticals was the sponsor for the initial phase 1 trial. They've been renamed Ionis Pharmaceuticals and entered into a partnership with Biogen. And so Biogen and Ionis are running this new trial.
Same group, Millers.
And Tim Miller is still the academic PI for this. Right, he's at WashU, and has done really, really commendable work in not just the trial, but also understanding the mechanisms at play and the biomarkers, and really done detailed work to sort of give a good landscape to give the trial and the treatment the best chance of being understood. Good question.
What would be the most effective thing I could say to a health insurance company if our doctor were to recommend Radicava for my husband's treatment, but the health insurance company denied coverage?
The question is, what's the best thing to say to an insurance company that denies coverage for Radicava, even when it's suggested by the healthcare provider? So, I think my approach, I think, will be multi-pronged. I think that having the care team and the person with ALS and the caregiver sort of all approaching this the same way. It is. I'm not sure we can do better than to say it's FDA approved. There is scientific, research saying that it's effective, and it is a new therapy, and there's a cost associated with that. But this is one of two potentially two effective FDA-approved therapies for a devastating disease, and I hope that will be enough to get us to move the needle. But I also hope that the work that ALS Association is doing, that MT Pharma is doing, and that clinician groups are doing now to impact insurance companies now will keep us out of that situation. But Barb has a...
Let me just add to that by saying that in our meetings with MT Pharma, they have approached at least 20 different health insurance providers and are receiving positive responses to that. So right now, MT Pharma is working through the system that they need to work through to be helpful on that front. So I totally agree with what Jim has said in terms of the convergence, but I also think that that you're going to start to see MT Pharma putting out information, more information about this as it gets closer to the time that the drug's available, and as they finish up negotiations with healthcare insurance companies. And I'm going to go out on a little limb, and I know I'm going to get staff back in DC that'll smack me around, but I'll commit to you all that as we know more about that we will be updating our frequently asked questions online, and we'll work with chapters on language that we can use, that we'll work with the good docs like Dr. Berry and others on developing some template language that we can use and that you can use as family members when you're on the phone with them. And you could say this. So we'll work together to make sure that we all are using the same language that's going to get the most powerful result. Okay?
Yeah, Dr. Perry, on this subject, it might be difficult for a patient to be able to justify getting Radicava unless we had some way to say they're in this group or that group, or CPK's high, or whatever. Some way that the prescribing physician can say, hey, this is the type of patient that benefits. So that's also part of the type of information you need to get out, is we got this drug, maybe it won't be approved just by asking, but hey, this patient has these signs and symptoms and blood levels, and so it's a perfect place for it should look.
Yeah. So I think this is a very good point. One of the things that happens after a drug is approved like this is we don't have all the answers about it yet. And we hope that we will have more of these answers over time scientifically. And the way that's going to happen is, one of the ways that that'll happen is this. The drug is approved. We have ongoing observational studies where people give blood and spinal fluid to be a part of the fight against this disease, to be a part of marching research forward, and many people in the room may have participated in research like this. We will have research studies that span the time period pre and post Radicava, Edaravone. And they may hold clues to help us analyze biomarkers, just like you talked about, to say there are some people that respond better or not. It may be difficult to say that there is a subgroup that responds better or not. And in fact, you know, the fact is, this same debate has raged on about riluzole for a long, long time. And ultimately, more and more evidence sort of showed that in every trial, though it wasn't randomized around riluzole, when we look at people on riluzole versus off riluzole, we see again and again and again that there's an effect of riluzole. It's not everything we hope for. It's a modest effect, but it's an effect. And it doesn't seem to be a small subgroup of people, but rather sort of across the disease. And so, you know, that's kind of how that drug is typically used now. And it's less of a debate than it was 15 years ago. But I think we need to, you know, that's why this period is both incredibly exciting to have a new treatment and also a real opportunity to understand the disease better and where to take drug development forward as well. That's a great question.
This is a real layman's question, so I apologize. But all of the studies seem to be kind of fighting inflammation. That word kept coming up a lot. So, you know, is there any benefit into doing kind of, you know, traditional things to fight inflammation, like foods and anti-inflammatory foods and, you know, the off-the-shelf stuff? Or is this a totally different inflammation?
That's a very good question. So the question is, many of the trials that I talked about centered around inflammation, and is there any sense in using sort of anti-inflammatory foods or manipulating diet to try to reduce inflammation? And I'll start with the broad truth is, which I'm not sure, but let me give you a little more detail on this. Until about 7 or 8 years ago, there were very few researchers that believed that inflammation existed in ALS. Stan Appel at Methodist was a strong believer over the decades and sort of carried that flame forward. And it was very difficult to get funding to look at inflammation in ALS because broadly it is not a neuroinflammatory disease. When you look at the spinal fluid, you don't see inflammatory cells in the traditional sense. This is not a disease that like other neuroinflammatory diseases, is sort of abundant in inflammation. But as we applied new tools to look very carefully, there is neuroinflammation in a very specific way. And so there have been a number of trials looking at anti-inflammatories and even looking at sort of transplant regimens to knock down the immune system. They have not broadly been effective. And so we think we maybe need to take a more nuanced approach to really target the kind of inflammation we're seeing here. So I would start by saying I think just broadly, knocking down the immune system probably isn't helpful. Whether any of the kind of dietary changes that one might do to try to reduce inflammation are on target or sort of more broadly effective is an even more complex question. So I don't know the answer to that, but I think it bears thinking about. And I guess my one caution around food is this. We know from looking at databases that people who have a stable weight have slower disease progression. There's some evidence from mouse models that if you overfeed mice, that slows their progression. In people, it's harder to come by evidence. There's some small studies, harder to come by broad evidence to say we can slow progression by, maintaining weight. But from the evidence we have, if you were going to be in one group or the other, you'd want to be in the weight stable group. And so I just always caution people when it comes to diet about not being too restrictive so that it leads to weight loss. But beyond that, I think it's a really interesting idea. I think you.
Maybe just the one more.
Oh, okay.
Sorry.
I'm going to be around afterwards too,
Yes.
so I can answer.
Aren't there clinical trials for C9? And how do you get on a clinical trial? Who decides what to do and how to do it?
Yeah, this is a great question. So this is a two-part question. One is, are there trials for C9 ALS, ALS that's caused by a genetic mutation in the C9orf72 gene? And then the second question is, how do you get into trials? Who decides? How do you find them? So let's take the first one first. So C9 mutations cause about 5 to 10% of ALS. Depending on where you are in the world, much higher percentage. So we talked about the SOD1 antisense oligonucleotide as a partnership between Ionis and Biogen. That same partnership is developing an antisense oligonucleotide for C9 ALS. That's in that sort of active preclinical phase. There is hope for a trial in the not distant future that's specific to C9 ALS. And there are gene therapies that are in development as well for C9 ALS. So I think it's an incredibly active area of research, and we're hopeful that soon we'll have a trial. The second question is and this is, I think, a good question since this is the last one we can do. I think it's a good question to end on. How do we find out more information? How do we think about enrolling in a trial? Number one, you can always talk to the physician, the multidisciplinary team, the physician you're seeing for treatment. The ALS Association has webinars about this, and you can go to their website. The Northeast ALS Consortium has webinars about this. And there are a few, the ALS Association also has a place to look for trials, as does the Northeast ALS Consortium, which goes by NEALS. This is actually started here, but it's a nationwide network now, even expanding internationally. So neals.org is one place you can go. And then there's a clearinghouse that's a little hard to use, but has all the clinical trials in it, called clinicaltrials.gov. I think that the attempt at the NEALS website is to organize the clinical trials in an easier to use format. And so you can find what sites the trials are happening at, you can find contact information, and you can find out about whether they're in an enrollment phase or not. Every trial has inclusion criteria. We talked about those with regard to kind of who was included in the Edaravone studies. And so there is a more detailed conversation that happens after you open the conversation of, are there any trials enrolling at my center? Is there a nearby center I can go to? Typically, people have to go to the trial center for the outcomes to be done. So you might have to look for something that you can get to, whatever that distance is. Thank you. Absolutely.